Hi, welcome to my YouTube channel Bio Explorer. I am Dimut Jayaratna. This is an educational channel and it will be very useful to improve your biological knowledge. Today in this video we are going to talk about another main organic compound found in living organisms. It is protein. Proteins. They are another main organic compound found in living organisms. Proteins play different functions in the body. They are made up of amino acids. A protein is composed of one or more linear chains of hundreds of different amino acids combined by peptide bonds. Therefore, it is called a polypeptide. 20 different amino acids are involved in the formation of proteins. Main elemental composition is carbon, hydrogen, oxygen, nitrogen and sulfur. All amino acids have a basic structure which is composed of a central carbon atom, an amino group, a carboxyl group, a hydrogen atom, and a variable group symbolized by capital R. R group is also called the alkyl group. It determines the identity of the amino acid. The R group is also called the side chain, differs with each amino acid, whereas the other groups are in the backbone, including hydrogen atom. At the center of the amino acid is an asymmetric carbon atom except in glycine. In the case of glycine, R is replaced by hydrogen atom. The central carbon atom becomes asymmetric when 4 different groups are attached to it. Amino acids may consist of one or more carboxyl groups and amino groups. Amino and carboxyl groups are shown to be neutral for ease of study. But typically, amino group is protonated and has a positive charge, while carboxyl group is deprotonated and has a negative charge. Therefore, amino group has alkaline nature and carboxyl group has acidic nature. When both characteristics are found in one molecule, they are known as amphoteric molecules. Therefore, amino acids are amphoteric. The properties of the side chain determine an amino acid's chemical behavior, that is, where it is considered acidic, basic, polar or nonpolar. The properties of the side chain determine an amino acid's chemical behavior, that is, where it is considered acidic, basic, polar or nonpolar. Examples for nonpolar and hydrophobic amino acids are valine and leucine. Examples for hydrophilic and polar amino acids are serine and glutamine. Now, let us see how the amino acids actually connected together in a polypeptide chain. Two amino acids undergo condensation reaction and form a covalent bond known as a peptide bond. Here, a water molecule is released. Therefore, this reaction is also called dehydration synthesis. This water molecule is formed by the hydroxyl group in the carboxyl group of one amino acid and the hydrogen atom of amino group of the other amino acid. Polypeptides differ from one another by the chemical properties and order of amino acids. At one end, the polypeptide has a free amino group, and at the other end, there is a free carboxyl group. Protein is composed of one or more polypeptide chains which are composed of amino acids. We can identify certain levels of proteins according to the structure and functions performed by those proteins. Levels of protein structure. There are 4 levels of structure which play important roles in their functions. Primary structure, secondary structure, tertiary structure and quaternary structure. First one, primary structure. This is the simplest level. Unique sequence of linearly arranged amino acids linked by peptide bonds is the primary structure of proteins. Each polypeptide chain has its own order of amino acids. The sequence of amino acids is determined by the DNA sequence of a particular gene that encodes the above protein. Secondary structure. The primary structure of a single polypeptide chain coils and folds to form the secondary structure. These folded structures are formed due to the intermolecular hydrogen bonds between the oxygen atoms of carboxyl group and hydrogen atoms attached to the nitrogen atoms in the amino group of the same polypeptide chain backbone. Backbone is the polypeptide chain apart from the R group. The most common types of secondary structures are alpha helix and the beta pleated sheet. Examples for alpha helix is keratin and for beta pleated sheet is spider's silk fiber. In alpha helix, a helical structure like a curled ribbon can be identified. Here, the carbonyl oxygen of the first amino acid forms a hydrogen bond with hydrogen attached to amino group of fifth amino acid and so on. The R groups in the helix are facing outward and free to interact with other molecules. 2 or more segments of a polypeptide chain line up next to each other to form a beta pleated sheet. Those segments are held together by hydrogen bonds formed between carbonyl and amino groups of the backbone. R groups can be seen on either side of the sheet. Many proteins consist of both alpha helices and beta pleated sheets, and some are less likely to be found in both ways. Tertiary structure. Secondary polypeptide chain bends and folds extensively to form the tertiary structure. It is a precise, compact, unique, functional and three-dimensional shape of the protein. Various interactions between the side chain R group of amino acids contribute to this structure. Those include hydrogen bonds, disulfide bonds, ionic bonds and van der Waals interactions. They are also called hydrophobic interactions. Generally, R groups with opposite charges form ionic bonds. Disulfide bonds are covalent bonds formed between sulfur-containing side chains. Hydrogen bonds are formed between polar R groups. Hydrophobic nonpolar side groups can form van der Waals interactions. Examples for tertiary structure are most of the enzymes, myoglobin and albumin. Quaternary structure. Quaternary structure of a protein consists of 2 or more polypeptide chains. Each polypeptide chain is called a subunit and involved in the formation of one functional proteins. Those subunits are held together by inter- and intramolecular interactions such as hydrogen bonding and London dispersion forces. But many proteins are made of a single polypeptide chain and have three levels of structure. Hemoglobin is a protein found in the red blood cells. It helps to carry oxygen and carbon dioxide throughout the body. Hemoglobin is made up of 4 subunits. Two of them are alpha type and others belong to beta type. Denaturation of proteins. Denaturation of a protein is the loss of a specific chemical three-dimensional shape due to the alternation of weak chemical bonds and interactions. This causes the protein to turn back into an unstructured chain of amino acids. Here, primary structure of the polypeptide chain will not be disrupted. The protein will lose only its higher order structures. Denatured proteins are usually non-functional. Sometimes denaturation can be reversed and sometimes it is permanent. Agents affecting the denaturation: high temperature and high energy radiation, strong acids, alkaline and high concentrations of salts, heavy metals, organic solvents and detergents. Functions of proteins. We can identify various types of proteins and let us look at some examples. Catalytic protein. They perform biochemical reactions. Examples are pepsin. It helps to hydrolyze proteins to small polypeptides. Amylase. Salivary amylase helps in the digestion of polysaccharides into smaller polysaccharides and disaccharide maltose. Pancreatic amylase catalyzes the conversion of polysaccharides into disaccharides. Structural proteins. They help to prevent desiccation. Examples are keratin. They are found in feathers and nails. Collagen. It is a major component in the extracellular matrix of animal cells. It provides strength and support to the cell. Storage protein. Examples are ovalbumin. It is the storage protein in egg, and casein. It is the storage protein in milk. Transport proteins. Hemoglobin. It transports oxygen and carbon dioxide. Serum albumin. It transports fatty acids. Hormones. They are long-distance signaling compounds and they are released to the blood by endocrine cells and organs. Hormones control various processes and functions in the body. Sometimes hormones are steroid-based and others are proteins. Protein-based hormones are called peptide hormones. Examples: insulin and glucagon. They are produced in pancreas and helps to regulate blood glucose level. Contractile motor proteins. Actin and myosin. Those proteins involved in the contraction of muscle fibers. Defensive proteins. Immunoglobulins. They eliminate foreign bodies. So that's all today. Hope you enjoy this video and gain a lot of facts about proteins for your biological knowledge. So please subscribe this channel and click the bell icon. See you in the next video.